Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Syami bin Razi Salwa from AS257. Today, I will present to you my FOP2 project titled Antimicrobial Resistance and Characterization of Possible Coagulous Negative Staphylococcus isolated from the raw milk samples at a selected farm in Alur Gajah, Melaka. There is an increasing worry about antimicrobial resistance worldwide, and the emergence of bacteria that are resistant to drugs in animals used for food production has become a notable public health matter. Raw milk often contains coagulous negative Staphylococcus species, which are responsible for causing bovine mastitis. There are three objectives that we want to achieve. First, to isolate the coagulous negative Staphylococcus and identify the antimicrobial pattern of cons isolated from the raw milk in Alur Gajah, Melaka. Second, to detect the biofilm formation by cons from the raw milk samples. And lastly, to identify the cons species from the raw milk samples using molecular identification. For the problem statement, high antimicrobial resistant and virulent genes pose a risk for farmers and veterinarians requiring effective therapies. Other than that, Staphylococcus bacteria species can form biofilm in slime, organizing into multicellular layers, enabling resistance to antimicrobial agent and host immunity, making the treatment intervention challenging. The significance of this study is that it can be used for further investigation about characteristic and virulent factors of cons that are related with mastitis. Other than that, it can determine the prevalence and antimicrobial resistance profiles which may have implication for the treatment and management decision. The widespread use of antibiotic in dairy farming has led to emergence of resistant bacteria strain. A study in Malaysia found a high prevalence of cons in dairy cattle, with most isolated exhibiting resistant to penicillin and tetracycline. Bovine mastitis is an inflammation of the mammary gland, primarily caused by bacterial infection with both gram-positive and gram-negative bacteria involved. However, cons, which is a group of opportunistic pathogens, have gained increasing attention due to their association with subclinical and clinical mastitis, which causes substantial losses in milk production, reduced milk quality, and also increase their cooling rates. For the methodology, first we went to a selected farm in Alur Gajah, Melaka and were able to collect 12 cow milk samples. During the collection, we also performed the California mastitis testing to detect the subclinical mastitis in cows. Then we isolate the cons from the milk samples on the blood agar and mannitol salt agar until we got a pure colony. After that, we proceed with gram staining and biochemical tests, including catalase and coagulase tests, to confirm the cons species. Next, we perform the antimicrobial susceptibility testing to characterize the isolate based on their pattern of antimicrobial resistance. Moreover, we also conduct a biofilm formation assay to detect the biofilm formation by cons from the raw milk samples, along with 16S rRNA gene sequencing to identify the cons species related with subclinical mastitis. Now we move to the result and discussion. A total of 12 cow milk samples was collected at Alur Gajah, Melaka. The CMT was done during the sampling, and 9 out of 12 cows showed of having mastitis. For the isolation process, 1 ml of milk sample was cultured into 9 ml of TSB and incubated overnight at 37 degrees, and then plated onto the mannitol salt agar to selectively isolate Staphylococcus species from the mixed cultures. The desired coagulous negative Staphylococcus colony was selected from the mixed colonies and cultured onto the MSA and also blood agar to get a pure colony. The morphological characteristic of these colonies was then observed. The pinkish-white colonies on the MSA were presumed to be cons with no hemolytic reaction on the blood agar, while the yellow colonies with a surrounding yellow medium on MSA and beta hemolytic on the blood agar were presumed to be coagulase positive, which is Staphylococcus aureus, thus excluded from this study. After that, we performed a gram staining and all the isolate were observed to be gram-positive cocci in cluster. To confirm that the isolates were indeed cons, biochemical tests including the catalase test and coagulase test were conducted. The result of biochemical tests revealed that they all show positive result in catalase test while negative result in coagulase test. Therefore, we will confirm that the isolates were coagulase negative Staphylococcus species. Then we proceed with antimicrobial susceptibility testing. The results indicate that cons isolate were most susceptible to gentamicin, erythromycin, and vancomycin, followed by ampicillin and tetracycline, and finally clindamycin. Meanwhile, they were highly sensitive to penicillin and oxacillin. The cons isolate were the most resistant to penicillin, which was in agreement with previous studies in China, which the resistance to penicillin was the highest, followed by the oxacillin by cons. Finally, there were 3 isolates were multi-drug resistant, while the rest of them were at least resist to one antimicrobial. The reason for those isolates to be MDR in the study is not clear. However, it may due to the imprudent utilization of antimicrobials. Study done by Ibrahim in 2022 mentioned that majority of mastitis causing cons like Staphylococcus epididymitis and Staphylococcus xylosus demonstrate resistance against various of antibiotics, specifically beta-lactams and tetracyclines that are frequently utilized in veterinary practice for mastitis treatment. For biofilm formation assay, based on the result, 3 isolates were biofilm former while other 4 isolates were non-biofilm former. Tremblay in 2012 observed that dairy calves with mastitis had a significant proportion of biofilm formation in their central nervous system with a percentage of 96.7%. The low percentage of cons biofilm formation in this study could be due to the result of using different growth media. In this study, we employed TSB for biofilm development experiment, whereas Tremblay used a brain heart infusion. Among the biofilm former isolates, 2 of them were moderate biofilm former and only 1 isolate was weak biofilm former. This could be due to the various constraints have varying capacities for biofilm production. Based on research by Saline in 2020, 2 of the most studied factors are known to be strongly correlated with biofilm formation, including the biofilm-associated protein and the product of ikaoperon, which regulate the synthesis and export of the most significant adhesive molecule of staphylococcal biofilm, the exopolysaccharide intracellular adhesion (PIA). However, the low percentage of biofilm development in our study implies that the production of biofilm by cons is not widespread in subclinical bovine mastitis. Thus, more studies need to be done to further explore the molecular epidemiology of biofilm formation of cons from the subclinical mastitis cases in dairy cattle. For molecular identification, UMMP84 and UMMP222 isolates were selected as they had the strongest antimicrobial activity and biofilm forming ability based on antimicrobial susceptibility testing and biofilm formation assay respectively. Both isolates managed to be extracted with analysis of DNA concentration and purity followed by the PCR assay and purified PCR product. The purified PCR product was sent to a third-party service, NextGene, for 16S rRNA sequencing. Unfortunately, we were not able to obtain the cons species from the raw milk samples as the blast nucleotide sequence revealed that the isolated UMMP84 and UMMP222 has 93.61% and 92.5% similarity percentage respectively with the Macrococcus casulacticus species. We were not able to differentiate between Macrococcus species and cons species due to the similarity of characteristics and both are part of Staphylococcal family. For example, both of them are coagulase negative and catalase positive. In summary, we failed to obtain the coagulase negative Staphylococcus species. Instead, we got the Macrococcus casulacticus species based on molecular identification. Moreover, the ability to form biofilm was rare among the Macrococcus casulacticus species isolates studied in this study, and the majority of the Macrococcus casulacticus in this study were resistant to penicillins and oxacillin. In the future study, this research can be improved by adding other tests to differentiate Macrococcus species and cons species, such as Voges-Proskauer test. In addition, the relationship between antimicrobial resistance and biofilm formation in Macrococcus casulacticus in this study is still not clear. Thus, this concept needs further investigate, including the genotyping study associated with biofilm and antimicrobial resistant genes in the future. That's all from me. Thank you.